about a protein purification technique, that is, or enzyme purification technique, which is dialysis. In this method, small molecules are separated from large molecules by allowing diffusion of only small molecules through selectively permeable membrane. In this process, the solution is put inside a semi-permeable bag, as you can see in this picture. The solution is inside the semi-permeable bag and is also suspended to a solution of low ionic strength. Due to the diffusion of the material from the bag to the hypotonic solution, after some duration, we have the pure material inside the bag, which is the enzyme, which is then subjected to ion exchange chromatography for further purification. So the enzyme or the solution containing the enzyme is put inside this membranous bag and kept or suspended in a solution of low ionic strength. So after some time, diffusion will occur and we will get the enzyme inside the bag, which is further purified by ion exchange chromatography. Now, for this purpose, the requirements are dialysis membrane, sodium carbonate, glass beaker, magnetic stirrer, dialysis buffer, macromolecule containing sample to be dialyzed, and clamps. Now, how to activate the membrane? 8 cm dialysis membrane to be used. Placing the membrane in 50 ml of 30% sodium carbonate. Boil for 10 minutes. Wash it in boiling distilled water for 10 minutes. This is the procedure by which we activate the membrane before it is filled with the solution containing the enzyme. Now, how the procedure of this is the procedure of dialysis. Let's discuss. Take out the dialysis membrane from the sodium carbonate solution, that is activation procedure, and rinse with distilled water. Knot one end of the membrane with a double knot. Fill the membrane with macromolecule containing sample. Knot the open end. Immerse the membrane in a beaker or flask containing large volume of desired buffer and keep it overnight at desired temperature. Remove the dialysis bag membrane from the buffer. Hold the membrane in vertical position. Sample to be removed using pasture pipette. So it is like after activation of the dialysis bag or the membrane, we have to take out the membrane from the sodium carbonate solution, and then we have to rinse this membrane with distilled water. After that, we have to knot the membrane, knot one end of the membrane with double knot. Now after that, the other end is filled with macromolecule containing sample. After that, knot the open end. So the sample is inside the membrane. Now immerse the membrane in a beaker or flask containing large volume of desired buffer. So in the buffer solution, it is suspended and kept it overnight at desired temperature. Now after that, remove the dialysis bag from the buffer. Hold the membrane in vertical position and sample to be removed using pasture pipette. So this is how dialysis is done.